Melanomas are tumors of cells that produce melanin, the pigment that protects our cells from UV radiation. These cells, called melanocytes, are found in the skin, but they're also found in the eyes, the inner ear, and mucosal surfaces. Although the exact mechanism remains unknown, we think nearly all skin melanomas, also called cutaneous melanomas, are caused by exposure to UV radiation. UVR can cause cell mutations and DNA damage that leads to tumors. The greatest source of UV radiation is sunlight. The UV radiation that reaches the Earth is made up of 95% UVA, 320 nanometers, and 5% UVB, 290 nanometers. UVA penetrates more deeply into the skin and can be more easily reached by the melanocytes. Both UVA and UVB have a potential role in causing melanoma. This year, laboratory investigation presented 2 special issues with new and exciting breakthroughs in melanoma investigation and timely overviews of the topic. Studying the causes of melanoma in humans is difficult because of the large time gap between UV exposure and tumor development. Day and others review the usefulness of mouse models to explore the role of UVR in melanoma formation, the protective effects of sunscreen, and the importance of melanin. Although UVR seems to be a key factor in melanoma development, UVB is actually essential for vitamin D production in the skin, providing over 90% of what our bodies need. Slominski and others review the double-edged nature of UVB in melanoma and explore the role of vitamin D, which is involved in important cell processes, regulate cell apoptosis, and can attenuate tumor progression. The other big mystery in melanoma research is that it can develop in areas of the body protected from UVR, like the palms of the hands, the soles of the feet, and under the nails, known collectively as acral melanomas. A review by Merkel and others looks at the features of these non-UVR melanomas and discusses potential mechanisms of mutagenesis. In another paper, Rawson and others analyzed the genetic mutation signatures of acral and cutaneous melanomas using data from the Australia Melanoma Project. The study shows that the nail plate and acral skin do not provide complete UV protection and confirms that cutaneous melanomas not caused by UVR are rare. Accurate diagnosis of melanoma is vital for patient outcomes, but is complex and subjective. Titus and others investigate this in a survey of pathologists to determine the influence of factors like age and tumor regression on the severity of diagnosis, and find that older patients tend to get more severe diagnoses. Classification of melanoma has moved on to also include the genes thought to drive the tumor. For example, Kiuru and others look at the NF1 gene, which normally acts as a tumor suppressor. Mutations in this gene cause a disease called neurofibromatosis type 1, predisposing carriers to tumors. Targeting the pathways regulated by NF1 could offer therapeutic options for treatment of melanoma. Once a tumor has formed, it has the potential to metastasize, to migrate to other parts of the body. The precise mechanisms remain unknown, but in a review, Moy and others look at the ability of tumor cells to invade lymphatic vessels and to migrate along the external surface of blood vessels without entering them, known as angiotropism. Other papers in this issue explore different mechanisms and enablers of metastasis, the anti-metastatic effects of certain drugs, and the regression of tumors in primary cutaneous melanoma. Understanding the pathways that lead to melanoma is important. In a review by Fisher and others, the authors look at the master transcriptional regulator of melanocyte lineage, MITF. This transcription factor has important cellular functions, but depending on the gene's mutational status, it can stimulate pigmentation, which shields from UV, or it can lead to depigmentation and even function as an oncogene. MITF could be an attractive target for melanoma therapy. Aside from the DNA and chromosomal alterations in melanoma, the changes in the non-coding RNA are poorly understood. MicroRNAs are small, endogenous non-coding RNAs that regulate gene expression by blocking transcription. Bapapore and others sequence RNAs from invasive melanomas and define a distinct set of microRNAs associated with melanoma. Inhibiting oncogenes, which have the potential to cause cancer, is a successful way to treat melanoma. In a review by Mandala and others, the authors suggest these treatments also impact the immune system, altering the expression of immunomodulatory molecules. Combining such treatments with immunotherapy could be an effective way to fight melanoma. Despite these advances, melanoma often develops resistance to treatment. Other papers in this issue try to understand the mechanisms which allow this, so that we can treat melanoma more effectively in the future. In the end, we are only at the cusp of understanding and defeating this disease. However, in the past several years, real progress has been made, and investigative pathology has been a key part in the fight against melanoma. To read the Laboratory Investigation Special Issues on Melanoma, visit